In the coldest parts of the world, such as around the Arctic, there is a layer of earth more than 1,000 feet deep in some places that will stay frozen solid all year round, known as permafrost. It behaves like a natural freezer, preserving in time the decomposition of plants and animals. In August 2016, a sudden outbreak of disease occurred in Siberia, with 90 people hospitalized and 1 child dying. Russian officials claimed it was caused by an infectious bacterium named anthrax. Scientists soon identified that the outbreak had started from an infected frozen reindeer carcass, which had recently thawed. It is believed that global warming has accelerated the melting of the permafrost, causing land instability and once preserved plants and animals to decompose, not only releasing gases into the atmosphere, but, according to scientists, also deadly bacterial spores. Of more concern is the possibility that not only known pathogens are being exposed, but also other forms of serious infectious bacteria, contagious viruses previously preserved in the ice. Could thawing permafrost unleash long-extinct deadly diseases? In 2017, a team of scientists specializing in the study of permafrost retrieved samples of organic material for testing from the world's most northernmost settlement, on the island of Svalbard in the Arctic Ocean, located halfway between Norway and the North Pole. They found that temperatures in the region are increasing at around twice as fast as the rest of the planet, and causing the permafrost to melt more rapidly. It was observed that the temperature of the permafrost had increased over the past 2 decades between 0.5 and 1 degree. Fortunately, not all bacteria and viruses survive once thawed out, and international law bans scientific study to prevent reactivating the most dangerous strains, such as smallpox, the Spanish flu, and tuberculosis. In the town of Longyearbyen, on the Svalbard archipelago in Norway, a cemetery has specially marked graves for those who died in 1917 and 1918 from the Spanish flu pandemic, which killed over 20 million people globally. Today, due to climate change causing increased ground instability, it is feared that these tombs could resurface due to landslides. The potential threat of releasing dormant Spanish flu is somewhat unknown. A team of curious scientists exhumed some of the bodies in the 1990s, but apparently they were unable to find any traces of the virus. Still, as a precaution against infection since the 1950s, the area around the archipelago in Svalbard does not allow burials to take place, but instead the dead are transported onto the Norwegian mainland. What really is the danger of unknowingly exposing yet-to-be-discovered infectious diseases? So-called time-travelling viruses that we know nothing about and may even, as has been suggested, have caused the extinction of Neanderthal man after being revived and released into the atmosphere at that time. Other time-travelling viruses are linked to Tutankhamun's tomb. Stories of the mummy's curse, or King Tut's curse, surround the mysterious deaths of many who have entered the 3,000-year-old sealed burial chamber of the ancient Egyptian ruler King Tutankhamun, who ruled around 1400 BC. On February 16, 1923, in Thebes, Egypt, an English archaeologist, Howard Carter, first discovered the tomb, and over time several deaths occurred of various members of Carter's team and visitors to the site, inspiring speculation in the belief of a curse, alleged to be cast upon anyone who disturbs an ancient Egyptian mummy, especially that of a pharaoh. Inscriptions have been found inside tombs giving warnings. Cursed be those who disturb the rest of a pharaoh. They that break the seal of this tomb shall meet death by disease that no man can diagnose. This curse does not differentiate between thieves or archaeologists, and it is claimed to cause bad luck, illness, or death. Since the mid-20th century, many authors and documentary makers have speculated that the curse is real in the sense of having scientifically explainable causes, such as bacteria or radiation. Is it just a coincidence, for instance, that Sir Archibald Douglas Reed, responsible for the initial X-rays of Tutankhamun's body, got sick the next day and died just 3 days later of a mysterious illness? A similar theory also surrounds the fate of archaeologist A.C. Mace, who took over further excavations after Howard Carter led the initial main dig, but the longer he stayed on site, the weaker he became. until he collapsed and died of suspected arsenic poisoning. Furthermore, within just 6 years of the discovery of the tomb, Carnarvon's 2 half-brothers, Carter's chief archaeologist, his personal secretary, his secretary's father, the excavation's radiologist, and at least half a dozen other prominent visitors to the tomb, were all dead. After the death of an American Egyptologist in 1995, politicians ordered a scientific study to investigate the possible hazards to health from so-called time-travelling diseases. It was suggested that a possible explanation for many of the deaths could be related to exposure to deadly strains of bacteria and mould on the mummies, but also from decomposed foodstuffs that could severely affect those with a weakened immune system. Recent laboratory studies have revealed that some ancient mummies do indeed carry mould, including at least 2 potentially dangerous species, Aspergillus niger and Aspergillus flavus. These moulds can cause allergic reactions ranging from congestion to bleeding in the lungs. Some tomb walls may also be covered with respiratory assaulting bacteria, like Pseudomonas and Staphylococcus, sometimes described as pink slime. Scientists have also detected ammonia gas, formaldehyde, and hydrogen sulfide. In strong concentrations, they could cause burning in the eyes and nose, pneumonia-like symptoms, and in very extreme cases, death. Due to rapidly melting permafrost in areas of Siberia, the Pleistocene period of the Ice Age, some 30,000 years ago, frequently exposes fresh organic matter and the remains of animals that once inhabited the area, such as the now extinct woolly mammoth. Microbes that were once dormant in the ice for tens of thousands of years are awakening, producing carbon dioxide and methane, both powerful greenhouse gas emissions, potentially contributing to global warming. Studies have provided evidence that soils frozen since the late Pleistocene period, predominantly located in Arctic and subarctic Siberia, do contain a wide diversity of microbes that can be revived upon thawing after tens of thousands of years. For instance, in the regeneration of a plant from a 30,000-year-old fruit tissue sample. In 2015, French researchers Chantal Abigail and Jean-Michel Claverie took a 30,000-year-old sample of Siberian permafrost and discovered 2 different viruses, Mollivirus sibericum as well as Pithovirus sibericum. These were reactivated and became infectious once again. Referred to as giant viruses, as comparably so large they can be seen under a microscope, laboratory tests demonstrated that these deadly pathogens multiply to attack single-celled organisms like amoebas, and therefore not those of humans or other animals. Research written in a paper published in the Proceedings of the National Academy of Sciences concluded, "We cannot rule out that distant viruses of ancient Siberian human or animal populations could re-emerge as Arctic permafrost layers melt, or are disrupted by industrial activities." Modern-day emerging microbial threats from melting permafrost in the Arctic have been shown to impact the local population, as it can affect the water and sewage systems, causing certain respiratory diseases. Conversely, the Arctic soil is also host to what is known as bacteriophagy, or viruses that kill bacteria. These viruses are important to medical science, as they have the potential to curb antibiotic-resistant bacteria. Each bacteriophagy targets a different bacterium, and has the potential to develop into new treatments for combating deadly pathogens. Is it any coincidence that the river Ganges in India, for instance, has for centuries been revered for its self-cleansing and special healing properties? More than 450 million people depend on the waters of the Ganges for many aspects of their lives. In 1896, one of the first published works on Ganges water by Ernst Hankin, a British bacteriologist, demonstrated an antibacterial property of Ganges water against the toxin cholera. Further work by French microbiologist Félix Derel in the beginning of the 20th century established that the antibacterial properties of Ganges water is due to a factor later termed bacteriophagy. The physical property of water sediment was observed in sampling and identified as locating from the headstreams of the Bhagirathi River, which flows from the melting permafrost of the Gangotri Glacier. With advances currently being made in genetic engineering, Could, for example, the Spanish flu pandemic of 1918 be resurrected as the ultimate bioweapon, or explored as a therapy to combat other deadly viruses? In 2003, Dr. Mohammed Majid, a cardiologist at the University of Texas Health Science Center, and Scott Lillibridge, a national bioterrorism expert and director of the Center for Biosecurity and Public Health Preparedness at the University of Texas School of Public Health in Houston, asked to consider the possibility of malicious genetic engineering to restore the 1918 Spanish flu strain. Sequencing of the genome of the 1918 Spanish influenza virus is nearly complete. Once it is published, unscrupulous scientists could presumably utilize candidate virulent sequences, they warn. All a future bioterrorist may need is something akin to a genome cookbook. Several factors favor influenza as an effective bioterrorism weapon. The virus is usually transmitted by direct contact, but it is also possible for airborne contagion, such as on a passenger plane. Influenza also occurs naturally, so initial clusters of intentional infections would be difficult to spot. With a relatively short incubation period, 1 to 4 days, a vaccine may not be an effective response following delayed recognition of a widespread flu virus release. An intentional release of a virulent flu strain could essentially result in a pandemic. We are unfortunately inured to the flu cycle, says William Schaffner, MD, chairman of the Department of Preventative Medicine at Vanderbilt University in Nashville. It would not cause terror. That's why it is not on the A-list of bioterrorism agents. But if our colleagues' attention to this heightens preparedness for pandemic flu, they will have done a good service, and I congratulate them. Majid emphasizes that viewing flu as a potential bioweapon only readies preparedness if another pandemic naturally occurs. Whatever thought we put into bioterrorism preparedness will be very useful for facing either a natural or man-made pandemic, he says. I hope there will never be another such a disaster. But since we know that more natural pandemics could be coming, it is a very timely and useful thing to do. Genetic sequencing work has been undertaken in part to try to understand the extraordinary virulence of the 1918 flu strain. A strand from the 1918 pandemic virus was isolated from a preserved lung tissue sample, and sequences found consistent with a novel H1N1 influenza virus that belongs to the subgroup of strains that infects humans and pigs. Another team of virologists determined that the 1918 strain was formed by a recombination of human and swine viruses that likely changed the virulence of the virus. To recreate a strain of the 1918 flu, if at all possible, would require enormous resources from a sophisticated laboratory, says William Bicknell, Professor of International Health at Boston University. It would absolutely have to be state-sponsored. More recent research, led by Dr. John Cash at the University of Washington in Seattle, studied what happened to the lung cells infected with the reconstructed H5N1 virus from 1918, with results suggesting that the deaths were the result of an immune system overreaction. We think the host's inflammatory response is being highly activated by the virus, and that response is making the virus much more damaging to the victim, Dr. Cash said. The host's immune system may be overreacting and killing off too many cells, and that may be a key contributor to what makes this virus more pathogenic. Scientists said the findings advise that focusing on the immune system would be key to tackling the bird flu virus. There have long been questions as to why the 1918 flu strain proved so deadly, and why so many victims were people not normally expected to expire from it. Similar patterns of mortality have been observed in those infected by bird flu, although the virus has yet to develop into a disease that spreads from person to person. Dr. Christopher Basler, a co-author of the study from Mount Sinai School of Medicine in New York, said the findings could help us develop more targeted therapies to combat pathogenic infections, including different types of influenzas, or perhaps avian influenza. Professor Carl Nicholson, the Professor of Infectious Diseases at the University of Leicester, said learning more about the cause of the high morbidity rate of H1N1 might then open the way for alternative methods of treatment. Could ancient unknown diseases and viruses be released by melting permafrost and cause a global pandemic? Let us know what you think in the comments below, and make sure to like and subscribe.